Welcome back to Zoology class. Good afternoon. Welcome to Zoology second year class. Already we completed the LAQs. Next, what about the SAQs means? SAQs also some we discuss in the first lesson the digestion and absorption. First A. Digestion and absorption. The important SAQs already I said one more time. I'll revise this one. The first one is the LS of tooth. Already yesterday only we discussed. I draw this diagram. Next is the second one is the protein digestion in stomach. Protein digestion in stomach. And the third one is the digestion and absorption of butter. Absorption of butter. Next is the functions of liver. These are the important SAQs in the first lesson, the first unit 1A, the 1A, the digestion and absorption. In that, these 4 SAQs are very, very important. The already almost these two we finished. One more time, I'll explain the protein digestion in stomach. With the equations, I will explain one more time. After that, I'll take the remaining 2. See, first. The protein digestion in stomach. Already I explained. One more time, I wait. Protein digestion in stomach. Normally, the proteins are not digested before that. Before that means the alimentary canal is having the mouth, buccal cavity, the pharynx, like esophagus, like this, they are present. But in that, the proteins are not at all digested. Protein digestion is started in the stomach only. Partially, the inside of the stomach, the protein digestion is occur. How the proteins are digested means the inside of the stomach, the gastric juices are produced. Here, inside of the stomach, the juices are produced. They are called as the gastric juice is produced. Gastric juice is produced. This gastric juice is having mainly the hydrochloric acid is present. The juice is having the hydrochloric acid. The mucus is present. Mucus is present, and also they are having the proenzymes. Proenzymes also present inside of this, and that the HCl activates the proenzymes, and the mucus is give the movement inside in the stomach. Clear? Next, I'll write it the equations how the proteins are digested. These equations are very, very important. The proenzymes means the gastric juice is having the proenzymes. They are the inactive enzymes. Clear? What are they means? The prorenin and the pepsinosin. Pepsinosin. Both of them are, these two are the inactive enzymes. They are the inactive enzymes. These inactive enzymes are activated by the hydrochloric acid. HCl activates the enzymes into active. Inactive enzymes are activated by active. That is the renin. Next is the pepsin. Pepsinosin is converted into pepsin. The renin is act on the milk protein. That protein is called as a casein. Here, Casein is the milk protein. This on the casein, which one? Renin, in the presence of calcium ions, form it as a calcium paracaseinate. Calcium paracaseinate. This calcium paracaseinate further form it as the further breakdown into calcium paracaseinate in the presence of next enzyme, that is the pepsin is in that in the presence of pepsin enzyme, they are converted into peptones. Means the milk is converted into peptones. Means the milk protein is converted into the peptones. Yeah. Next, the other than the milk and other proteins also there. That proteins are in the presence of pepsin enzyme converted into peptones and proteases. Peptones and proteases. Here, this is about the protein digestion in stomach, and this is the so the last class I'll draw the diagram of wellness of tooth. The second one is this one. Next, the butter digestion. Next, third question, third SAQ is the digestion and absorption of butter. Digestion. What butter is a fat? Actually, this is the fat. Fats are not digested in the stomach. The butter is mainly this one is a butter is a fat that is digested in the small intestine. Here, what it is means inside of the small intestine, the number of juices are produced. What it is means from the intestine, the inside of the intestine, intestinal juices are produced. Intestinal juice is produced. And the next, the bile juice from the liver, bile juice also enter inside of the small intestine, and also pancreatic juice. Inside of the intestine, these three juices are occurring. These mainly they are containing the enzymes. Here, mainly the bile juice and the pancreatic juice, both of them are helpful for the digestion of fat. How it is means the bile juice are having the bile salts are there. Bile juice is having the bile salts are present. These bile salts are emulsifying the fats. Here, what is the emulsification means the big macro molecules of fat is break down into micro molecules. That process is called as a emulsification. First, on the fat, bile juice is bile juice is containing the salts. That salts break down the fat into smaller micro molecules that we call as the emulsification. After the emulsification, the pancreatic juice producing the pancreatic lipase enzyme. Here, lipase enzyme is produced by the pancreatic juice. With that pancreatic juice, the emulsified fats are further digested as a fatty acids and glycerol. Here, how it is, I will write it here in the form of equation. Yeah, the butter means this one is the actually fat. This fat, how it is, the fat in the presence of bile salts. Bile juice is containing the bile salts. With that bile salts, they break down into smaller molecules that we call it as a emulsified fats. Emulsified fats. These emulsified fats further break down by the pancreatic lipase. Here, see this one here, emulsified fats in the presence of lipase enzyme, they are break down into monoglycerides. These monoglycerides further form it as the fatty acids and glycerol in the presence of lipase only. Monoglycerides in the presence of same lipase enzyme, they are formed as fatty acids plus glycerol. Here, this is about the digestion. Here, digestion is occur like this. Butter is the fat. The fat is in the presence of the bile salts, they are becomes as emulsified fat, smaller molecules. That on the emulsified fat, lipase is act and form it as the fat. The emulsified fats is converted into monoglycerides. The monoglycerides further divided into fatty acids and glycerol in the presence of lipase enzyme. This is about the digestion. Here, what about the absorption? Here, the forming the fatty acids and glycerol, how they are absorbed inside of the blood, we are going to discuss absorption. These are the fatty acids are they are not insoluble in the water. That's why they are directly they doesn't go to the directly to the bloodstream. First, they are formed as smaller molecules. Here, the fatty acids and glycerols, they enter inside of the mucosa of the intestine, and inside of that, they are formed as smaller molecules that we call as mucilus. Here, the fatty acids, glycerols are not directly, they are enter inside of the inside of the blood. That's why they are formed as they are formed as the mucilus, means they are break down into smaller molecules that we call as the mucilus. These are present inside of these are enter inside of the intestinal mucosa. These mucilus are enter into the intestinal mucosa. This intestinal mucosa is inside of the intestinal mucosa. These mucilus are covered by the protein coat. Here, mucilus are covered by, are coated by proteins. Protein coat is formed and form it as the chylomicrons. Mucilus are formed as mucilus are surrounded by the protein coat, form it as a globule-like structures. These globules we call as chylomicrons. These chylomicrons are absorbed by the villi. Here, the inside of the intestine, the folded structures are present. For example, this one is the intestine TS. Outer side serous layer is present. The inner side muscular layer. Next is the submucosa layer is present. The inner, like this, mucosa layer is present. The inner layer is called as a mucosa layer. In this mucosa layer is having the finger-like structures. They are called as a villi. Inside of the villi, lymphatic vessel is present. That lymphatic vessel is called as a lacteal. Here, this lymphatic vessel, inside of that lymphatic vessel, they can take the chylomicrons. They can absorb the chylomicron and given to the bloodstream. Here, these chylomicrons enter inside of the subclavian vein. Here, chylomicrons, they are enter into the bloodstream like a subclavian vein through the thoracic vein and the enter inside of the blood. And inside of the blood, they again, these chylomicrons are, see this, wait. The villi is taken the, which one? Chylomicrons. Here, they are taken the villi absorb the chylomicrons. These chylomicrons are enter into the subclavian vein, means the subclavian vein means they are the bloodstream through the thoracic vein. Thoracic vein, they enter into the subclavian vein, means they enter into the bloodstream. This chylomicron break down into, this is a breakdown into again the fatty acids and glycerol. Fatty acids and plus glycerol. Fatty acid only, not acids. Fatty acid and glycerol. Clear? Like this, absorption is occur. How they are absorbed? The fatty acids and glycerols are not directly absorbed inside of the bloodstream. That's why first they are converted into smaller molecules. They are called as the micelles. They are not the molecules. They are the droplets. Here, droplets-like structures are formed. The droplets are present enter into the intestinal mucosa. These droplets are covered by the protein coat and form it as the structures are that we call as lobules are formed that we call as pylomicrons. These pylomicrons are absorbed by the villi containing lactic acid, lymphatic vessel. Here that is called as a lacteal. Lymphatic vessel is present inside of the villi. That is called as the lacteal. This lacteal absorb the chylomicron and that chylomicron enter into the bloodstream like the subclavian vein and enter the bloodstream. They are again break down into the presence of lipase only. Again, in the presence of lipase enzyme, they are converted into the breakdown into fatty acids and glycerol. These fatty acids are absorbed by the are diffused into the adipose tissue by adipose tissue by adipocytes. Here, first the fatty acids and glycerol enter into the adipocytes and to the adipose tissue, or they are enter inside of the liver. Here, this is about the absorption. Here, proteins are digested as the fats means the butter is digested as a first inside of the intestine. They are digested as a fatty acids and glycerol. These fatty acids and glycerols are not directly absorbed by the small intestine. That's why they are first to form it as a micelles, like a droplet-like structures that is called as a micelles. They enter into the intestinal mucosa and they surrounded by the protein coat that lobules are formed that we call as a chylomicrons. These chylomicrons are absorbed by the villi lacteal. From the lacteal that goes to the subclavian vein like a bloodstream. Again, this one is break down into fatty acids and glycerols. They enter inside of the adipose tissue, either liver. Here, this one is about the digestion and absorption of the butter. Here, this one is the third answer. Next, we are going to discuss about the functions of liver. Fourth question, one is the functions of liver. The liver is present in the right side above the stomach. Here, the liver is located above the stomach. Right side it is present. It is weighing the 1.2 to 1.5 kgs. Here, the weighing of the weight of the liver is 1.5 to 1.2 to 1.5 kgs, and they are helpful in the synthesis. They are performing. The liver is performing more functions. They are called as a synthesis. More functions like a synthesis, secretion, and storage. Here, they perform main functions like a synthesis, secretion, and storage. Clear? What about this one means the first one? The function of the liver is the liver is producing the bile juice. Here, liver is producing bile juice. This bile juice is having the bile pigments and the bile salts. Major as the bile salts are present and the bile pigments are present. Here, the bile pigments are bilirubin and bilirubin and bilirubin wording here. And the main the salts are helpful. Salts are helpful in the emulsification. The bile salts are undergoes bile salts emulsify the fats here. Then only the fat digestion is occur. Otherwise, the fat digestion is not occur. Which bile salts mean sodium and the potassium, glycolates and tarcolates. Glycolates, and tarcolates. Tarcolates. Here, what is the use of this one means bile salts are mainly helpful for the emulsification of fats. Emulsification of fats. Next, second function. What is the function of the liver? The second means they play a role in the carbohydrate metabolism. It play a role in carbohydrate metabolism. Like glycogenesis, glyconeogenesis, glycogenolysis, sorry. Glycogenolysis, and gluconeogenesis. I'm writing here gluconeogenesis. Clear? The means play a role in the carbohydrate metabolism. Like this, just you write it as a like this only. But for the I'm explaining these three steps also. Just you write it as a in the functions. They play a role in the carbohydrate metabolism, like a glycogenesis, glucogenolysis, and the gluconeogenesis. Here, what is the glycogenesis means the formation of the glycogen. Here, from the glucose. From the glucose, the glycogen is formed that we call as a glycogenesis. Here, next the glycogenolysis means reverse. Here, the glycogen is breakdown and form it as a glucose that we call as a glycogenolysis. And what about the gluconeogenesis means glucose is formed by the amino acids from the other amino acids and lactic acid, the glucose is formed that we call as the gluconeogenesis. Like this, the liver play a role in the carbohydrate metabolism. Here, what is the first function? The liver secreting the bile juice that containing the bile salts and bile pigments. The bile salts are sodium, potassium, glycolates and tarcolates, and the bile pigments, bilirubin and biliverdin. Here, bile salts are helpful in the emulsification of fat. That is the first function. Next, the second one, it plays a role in the carbohydrate metabolism, like a glucogenesis, glucogenolysis, and the gluconeogenesis. Here, this is the second function. Next is the third function. Liver play a role in lipid metabolism. What is the lipid metabolism means? They synthesize the cholesterol and triglycerides. Here, they synthesize cholesterol and the triglycerides. Means liver is play a role in the carbohydrate metabolism as well as It play a role in the lipid metabolism also. Next, the liver is a detoxifying organ, means it help in the detoxification. Liver helps in the detoxification. Helps in detoxification. What is a detoxification means? They are removing of the toxic, means any poisonous substance along with the food enter inside of the body, that is eliminated. Toxins are eliminated by the liver. That's why we call it as a they are the help in the detoxification or detoxificating organ. Here, next is it is a thermoregulatory organ. Liver is it is thermoregulatory organ. Thermoregulatory organ. It maintains the temperature. Thermo means temperature. It maintain the temperature. That's why we call it as a liver is a thermoregulatory organ. Next is the it help in it play a role in it play a role in deamination. Amino group deamination play a role in deamination. And formation of urine through ornithine cycle. Through ornithine cycle. Here, it play a role in the means the deamination. It is mainly helpful in the deamination and also formation of the urine. Urine means amino group is converted into urine in the cycle through help of the cycle that is called as the ornithine cycle. Through the ornithine cycle, ammonia is converted into urine. Why the ammonia is converted into urine means ammonia is the more toxic, means more the concentration that is converted into less concentrated urine. That's why. Here, next it play a role in the deamination. Next, the lactic acid it converts lactic acid produced in the lactic acid produced in the anaerobic respiration. Anaerobic respiration. Converted into glycogen, converted into glycogen through Cori cycle. The help of the Cori cycle, what it is means the lactic acid is converted into the glycogen in the help of the Cori cycle. Here, this one is about the next one. Next, more the functions are there. Next is the 8th one. It acts as a liver. It acts as a hemopoietic organ. Hemopoietic organ in fetus. Means hemopoietic organ means they're producing the all the blood cells that we call the hemopoietic organ in fetus and the erythroclastic organ in adults. Next, means in the fetus, they act as a hemopoietic organ, means for the production of the RBC, WBC, and thrombocytes that is helpful in the fetus. And in the adults, they act as a erythroclastic organs, means the RBC is destruction is occur inside of the liver. That's why erythroclastic organ in the adults. Next, it synthesizes plasma proteins like albumin and globulin. Albumin and globulin albumin, globulin, anticoagulants, heparin, coagulants, fibrinogen, and prothrombin. Means it synthesizes as the plasma proteins they can synthesize, the anticoagulant they can synthesize, and also coagulants also they can synthesize. What about that one? It synthesizes plasma proteins means which are the plasma proteins? Albumin, globulins are synthesized by the liver only, and also the liver synthesize the anticoagulant like a heparin. The means they prevent the means they destroy the clot. That's why we call as a heparin is called as an anticoagulant, and the coagulants like a fibrinogen and prothrombin also the synthesized by the liver only. Next, last 10th one, the liver containing the larger phagocytic cells, they are called as a Kupffer cells. Clear? It has Kupffer cells. Larger phagocytic cells, they are larger phagocytic cells. What is the function of the functions of the Kupffer cells? Means they eliminate or they remove the unwanted the substances. Here, unwanted substances. And also microbes. Here, this is about the functions of liver. It is not only important for the exam point of view. As a human being, you want to know this one, the functions of the liver. The liver is a major organ for the functioning of all those things. Here, it plays a role in the synthesis, secretion, and also storage. One more time, all the together I explained, briefly I explained about the functions of liver. What it is, the first it is having it is producing the bile juice. This bile juice is having the bile salts. These bile salts are helpful in the emulsification of fats. Means they helpful in the digestion of fats. It is helpful in the digestion of fats. Next, the liver play a role in the carbohydrate metabolism. Carbohydrates are the digested, means carbohydrates are the breakdown by this liver only. Next, carbohydrate metabolism. And it also play a role in the lipid metabolism. Lipid metabolism means how it is, means the synthesis of the cholesterol and the triglycerides by this one only. That's why they play a role in the lipid metabolism. It is detoxifying organ. Detoxification, it help in the detoxification means the toxins are eliminated along with the food. Any toxins are enter inside of the body, that is eliminated by this only. Next, it acts as a thermoregulatory organ. Thermoregulatory organ means they maintain the body temperature. That's why we call it this is called as a thermoregulatory organ. And next, to help in the deamination. Deamination. Help in the, sorry. Deamination. Amino group is removed, and that is help in the formation of urine. Formation of urine, urea. Formation of urea through ornithine cycle, which are through the ornithine cycle. Next, lactic acid is converted into lactic acid in the produced in the anaerobic respiration. This lactic acid is converted into glycogen by the Cori cycle. Through the Cori cycle, they converted into the lactic acid is converted into glycogen. Next is the liver act as a hemopoietic organ. Hemopoietic organ in the fetus. Erythroclastic organ. Organ in adult. Next, liver is synthesized. The which one? They synthesize the plasma proteins. Plasma proteins, albumin and globulins. Plasma proteins are albumin and globulins, anticoagulant, heparin, and also coagulants, fibrinogen and prothrombin. Lastly, the liver containing the Kupffer cells are present. These Kupffer cells are the large phagocytic cells. They remove the unwanted substances and the microbes from the liver. This is about the functions of liver. With this, we completed the digestion absorption SAQs. We discussed about I draw the diagram last class. And the next, I explained about the protein digestion. And the third, the digestion and absorption of the butter, I explained. And the last, this one is the functions of liver. You learn it, these 4 questions. One will come compulsorily in the exam. Here, next is the next lesson. Sorry, B. Respiration. In this, the important are inspiration, mechanism of inspiration and expiration. Inspiration, expiration in the normal conditions. The process of the inspiration, expiration, normal conditions. And the second is the carbon dioxide transportation. Next is the respiratory movements. Next is the respiratory disorders. These are the important SAQs from the second lesson, the respiration. And first we are going to discuss about the first one, the mechanism of inspiration and the expiration. First one in the respiratory system, mechanism of . inspiration, and expiration. All of you know this one. The inspiration means taking air in. Expiration means expel the air out. Clear? This is undergoes a mechanism means the inspiration is the active process. The expiration is the passive process. How it is occur means in the inspiration mainly the lungs are present in the thoracic cavity. Here the thoracic cavity, just rough diagram. This one is the thoracic cavity, the lungs containing cavity. This cavity is the surrounding four sides, four bony structures are present. The back side, for example, rough diagram I'm drawing, the back side, the vertebral column is present, means the spinal cord, the not spinal, vertebral column is present. The front side, the chest bone is present, that we call as a sternum. This one is called as a vertebral column. This one is called as a sternum. And the sides above this one, the connected to the sternum and the vertebral column, the side bones are present. They are called as a ribs. Ribs are present. Next, this side, the back down means the posterior side of the thoracic cavity. The dome-shaped structure is present, that we call as a diaphragm. Here, like this, thoracic cavity is four sides, four type of bony structures are present. The back side means dorsal side, vertebral column is present, the ventral side of the thoracic cavity, the sternum is present, the posterior side, diaphragm is present. Above that one, the mainly the means lateral, the ribs are present. Here, with this only, with the inspiration, expiration mechanism is occur. Here, how it is, I'll draw the diagram. Afterwards, I will explain. See this one, the one on this side first, I'm taking the inspiration diagram, inspiration. This is expiration. Here, the inspiration. This is the pulmonary cavity. This is the vertebrae. Vertebral column is having the vertebrae backside. These are the rib cuts. Side means side bones, ribs cuttings. This one is. Here the diaphragm. Contract. Next, these are the raised ribs and sternum. Air comes in. Next, vertebral column. Next is the expiration. Here the diaphragm relaxed, comes dome shape. Dome shape. Next, this one is before that the raised ribs and sternum. Ribs and sternum comes original position. In this time, ribs and sternum. Otherwise I'll write it that side. Ribs and sternum comes original position. See, this one is the inspiration. This one is the expiration. First, we are going to discuss about the inspiration. Inspiration time, what will happen means in the inspiration, the expiration is mainly occur by the two types of muscles. They are called as a diaphragm muscles. Here, diaphragm is having the muscles that we call as a diaphragm. Having the muscles that we call as a phrenic muscles. No need the writing of the muscle name. Just the diaphragm muscles are participating in the respiration, and also the external intercostal muscles. External intercostal muscles. Where it is present means external intercostal muscles are between the ribs. For example, side bones ribs are like this. The ribs are having the muscles that we call as a external intercostal muscles. In the mechanism of respiration means in the inspiration and expiration, the two muscles are participating. One is called as a diaphragm muscles, and second one is called as a external intercostal muscles. Now, how the process is there means the inspiration is the active process. In this, what will happen means the muscles will be contract. In the inspiration time, the diaphragm muscles and the external intercostal muscles, both of them are get contract. The diaphragm muscles get contract. What will happen? Actually, diaphragm is a dome-shaped structure, but in the contraction time, they comes down. Dome-shaped is comes down. That time, the thoracic cavity is increased. The anterior-posteriorly, thoracic cavity will be increased. Next, in the ribs and sternum, ribs also the means the external intercostal muscles are contract. That time the ribs and the sternum raised means they comes to the up. That's why dorsoventrally thoracic cavity is increased. Means through the diaphragm muscle, the anterior posterior posteriorly cavity is increased. Through the external intercostal muscles, dorsoventrally the thoracic cavity is increased. Automatically thoracic cavity is increased means the what will happen? The inside of that, the lungs, the pulmonary cavity is increased. This one is a pulmonary cavity. Pulmonary cavity increased. Cavity space increase means automatically pressure will be decreased. That's why the atmospheric air is comes inside. That we call it as a the inspiration. Here means the contraction of the diaphragm muscle and external intercostal muscles. The pulmonary cavity will be increased. The pressure is decreased. That's why atmospheric air comes in. That is called as the inspiration. What about the expiration means that the thoracic cavity is decreased here comparatively here. The pulmonary cavity, thoracic cavity is decreased automatically. The pulmonary cavity also decreased. Why it is decreased means they are comes to the virginal position. Here diaphragm, the inspiration time they are becomes as a straight, but in this time they comes to the virginal position. That's why anterior posteriorly this decreased, and the ribs and sternum comes to the virginal position. That time dorsoventrally they are decreased. That's why the pulmonary cavity is decreased. The space is decreased. That's why the pressure is increased. That's why they comes out that we call as a expiration. Clear? The air comes out by the pressure inside of the pulmonary cavity. The pressure is increased automatically. The air comes expel out. Air expel out. Here, this one is about the expiration. In the expiration time, no need of the energy. That's why it is called as a passive process. This one is the inspiration is called as a active process. This one is active process. This is the passive process. Simple one. The mainly muscles are important. The phrenic muscle means diaphragm muscles, and the external intercostal muscles are mainly participating in the inspiration and expiration. The contraction time, inspiration is occur. The relaxation and comes original position time, they are formed as a expiration. Here, the next remaining SAQs means the 3 SAQs, more 3 SAQs are there from this lesson. I'll continue next class. Same time, next class is there.